Hello, everybody. I will give you an introduction on generative chemistry in real-world application, try to focus on challenges and opportunity. My name, as you have heard, is Jessica Lanini, and I am a data scientist working in the computer-aided drug discovery in Novartis, also known as CAD. But let me first give you, let's say, an overview where the CAD group really impact the drug discovery pipeline. So as you may have heard, the drug discovery pipeline is a very long and complex process. It goes from the discovering to the bringing the drug to the patient. And this, let's say, pipeline can take quite a few years, on average 14 years. And the CAD group actually impact the first segment of this pipeline, in particular in the context of lead discovery and lead optimization. So basically in the moment where we would like to discover novel compound and to optimize them in terms of different properties. Of course, the CAD tools have evolved over the years, and with the introduction of machine learning technique, we got a very boost in activity on our CAD group. Here I just mentioned some of the opportunities of applying machine learning in CAD. We have some example in retrosynthesis and activity prediction, in protein structure prediction. You may have heard about AlphaFold. Then automatizing the so-called DMTA cycle that I will present you later, or to come up with model for activity and off-target prediction. And as a center of this talk, they integrated de novo design. But why discover a novel compound is such a complex problem? This is, in the baseline, a searching problem, but the space where we are searching on is really big. It has been estimated to be 10 to the power of 60. It is a discontinuous space, and this problem becomes even more complex if you would like also to optimize with respect to specific property for which you need to have model. But most of the time, the data that you have available for building models for this property can be quite small and moreover biased. So in general, I would say that a drug candidate cannot be just identified, but needs to be designed. And the design is really the first part of what we call DMTA cycle. This is the classical process that you can find in each medchem project. We will start with the design, where basically med chemists would come up with some ideas, but this idea can come also from virtual libraries. And in the context of AI and machine learning, we also have the possibility to introduce novel ideas through generative chemistry. The AI is also, let's say, combined with some classical CAD tools related to physics-based technique that can help us, and structure-based design that can help us in creating this advanced digital workflow that can actually reduce the number of, let's say, ideas that we come up with in the first phase in order to prioritize the most promising result that can go into the make and test phase. In this context, also active learning is quite well used in order to prioritize compounds that are not only promising for the project itself, but also for the model perspective to improve the model later on. But what we mean by generative chemistry overall? So for us, generative chemistry is really a combination of components. We have a first component related to the molecule generator or the search space that, of course, can be derived from the simple enumeration, but in the context of AI will be mainly generative models. Then we have multi-objective optimization searching algorithm that would allow us to move inside this space. And these algorithms are normally based on searching functions that are based, of course, on properties that we would like to optimize our final compound. At the end, we can come up with the 100K solution that, of course, are too many for exposing this to the project team. So we need to really post-process such solution in order to obtain the most promising 100 or maximum 1,000 solution. Let me first go into the first part of this generative chemistry workflow, and in this case we are talking about generative models. The generative models architecture can change according to the featureization you would like to impose to your input data, that in the context of this talk is really the molecule. And one can model and can featureize the model, the molecule as a string, as in the case of the CDD generative models. This was introduced by colleague at Bayer, and they built this model, which is a translational autoencoder that is based on LSTM. And this is quite good, actually, not only for the generation, but also as a descriptor for property model. But maybe the most natural way of representing molecule is through graphs, because you can think of representing each atom of the molecule as a vertex of this graph, and each bond between the atoms as an edge of your graph. And this is the case of Molar, a model that our group has developed together with Microsoft Research in a collaboration, and I will go a little bit more in depth into that in the second slide. Of course, there are also other ways of featureizing the molecule. In this case, I wanted also to present you the PQSR2 compound model that was built internally in Novartis, and for this we used basically profiles on top of GANs. So let me go a little bit deeper into Molar. Basically, Molar was born for addressing a specific challenge that at the moment we had when collaborating with Microsoft in this challenging task of generative chemistry. The challenge was actually to fix a scaffold during generation, because in most of the cases when you work in a medchem team, you may have identified a chemotype of interest, so you want to keep a core of your compound to be fixed, and then maybe changing the exit vector in order to improve specific property of interest. And this is the case of Molar. Molar is a variational autoencoder based on graph neural network, whose main characteristic is that in the generation phase, it is not based only on atom level generation, but also on motif level generation, where with motif we mean the most common substructure that are, let's say, available. Indeed, they were built on top of, let's say, publicly available data. We can construct a real dictionary of motifs, and from this we can compute the probability distribution of having a specific motif during the reconstruction. And we can even impose to have the motif that we want at the end. As you can see, comparing with other generators, with the Guacamole benchmarking, which is a benchmarking tool for generative models, the Molar model works quite good in terms of different characteristics. Another challenge I would like to mention here is related to the second part of our generative chemistry workflow, which is related to building property model. Of course, you can think about many different models that can be used to build property models, many different architecture, hyperparameter, and in the context of, let's say, the chemical field, we have also a lot of different molecular representation, both traditional ones and data-driven ones. So we decided to build our internal AutoML tool, which is called Prefer, that is publicly available under RDKit, where we basically automatize the way of comparing different molecular representation and model type in order to find out what's the best combination for our use case. But unfortunately, this cannot address all the possible situations, and we can have specific cases where you need to maybe investigate a little bit further and build more sophisticated model, as in the case of small data. So this is quite common whenever you enter a project that you have a limited amount of data, especially for on-target and off-target activity. So we also explore, together with Microsoft Research, the capability of state-of-the-art models that can handle small data sets. So, for example, few-shot learning activities. And in this case, you can see that we have actually published a repository where we we can benchmark such model, and we have noticed that prototypical networks tend to perform much better with respect to the other state-of-the-art model against different number of training compounds. So this was quite promising and helped us to actually select the right model whenever we had a problem of small data. I just wanted to mention also another challenge that we face in the context of post-processing. When we do post-processing, we can, let's say, use a lot of different in silico tools, but also some filters, some, let's say, characteristics of the outcome that we would like to maybe not see, some chemotype that have some alert that we would like to exclude. But it's very tough to also include in this evaluation the expertise knowledge. So together with Microsoft, again, we also worked in the context of MolSkill. This is a score that has been learned by a psychometric experiment. Basically, we had some med chemists internal to Novartis that were exposed to 2 compounds and then were asked to select one of the 2 every time. And then we learn basically their selection over and over, and then we try to have a model that would proactively tell us that one compound would be better than another one. So this model is, this score actually is actually performing better than, for example, the QED score that basically quantified the drug likeliness based on property of the molecule. And you can see this basically in the context of having the possibility to distinguish between the distribution of Campbell compounds that are more, let's say, nicer for the med chemist, and the GDB17 from a virtual space compound that can be more exotic in a sense, while the QED was not able to really distinguish the 2. So we actually use it proactively during the, let's say, filtering procedure. Now, just a concrete example from the public domain, just to show you generative chemistry in action somehow. This is an example of EGFR. So EGFR is a tyrosine kinase targeted in non-small cell lung cancer, and we basically started by using specific drugs that were targeting EGFR with a specific core that we were interested in. In this case, it's the aminoquinosalin core. And for this specific target, we also have an X-ray structure. In this case, we have the EGFR bounded to such compound with the aminoquinosalin core. And the first step in order to implement your generative chemistry pipeline is really to start by building your local model. So we built our on target and off target activity model as a first step, evaluating the corresponding characteristics and quality against our temporal split data. And then we had the possibility to set up our MPO. So the multi-parameter optimization, you should really evaluate which are your models of interest, what you would like to optimize, and which substructure constraint you would like to keep, and so on and so forth. So we run the optimization, and we obtain 30K novel generated compound. Here you can see the evolution over the different optimization step of the different scores for CDD and molar. And on the right-hand side, you can see the property distribution of the top compound. You see them against, in red, the seeds compound, so the drug that I've presented at the beginning that are known to be, let's say, active against our target. Finally, I would like also to show you the distribution of this compound in the UMAP of the chemical space of EGFR, just to show you how we can maybe explore a region of the space and be more exploratory when we would like to be. And at the end, we always do a 3D constraint assessment, because in the generation itself, the 3D information is not included, so we will generate only according to properties and off-target and on-target activity. But since we have the possibility to access to the 3D structure of the target of interest, then definitely this last step is crucial to really identify the top compound. And to finalize, I would like to give you some take-home messages. So first of all, it's important to manage the expectation, because sometimes, especially when you present this to the medchem team, they get quite excited and they think that maybe in one iteration they will get the DC. Well, this is not the case. It needs a lot of iteration. There is no magic behind, and it needs quite some feedback. It's still in evolution, so basically these generative methods are new also from the academic field, so we always are, let's say, exposed to novel algorithm and novel strategy that can help us in improving also our pipeline, and we are also learning how to use it properly, how to set this correctly. And finally, the key for the success is really to integrate all of them. So integrate the best you can also from the traditional tools that can be leveraged with machine learning and this technique, and also collaborating as much as possible with the medchem team to have this work at the end of the day. Here I would like to acknowledge quite some people that are working in this challenging project that is in collaboration also with Microsoft Research. As I mentioned at the beginning, it was a long journey that started in 2019, and I would like also to thank you for your attention today.